Hello students, welcome back to my previous year question discussion series of the paper Animal Cell Biotechnology. The question is from University of Calcutta, year 2022 question series. Number 1, state the principle of RT-PCR carrying 2 marks. What is the use of ethidium bromide in gel electrophoresis carrying 2 marks? What is DNA microinjection carrying 2 marks? What is BAC carrying 2 marks? Distinguish between continuous and finite cell lines carrying 2 marks. State one use of airlift bioreactor carrying 2 marks. What are the disadvantages of using antibiotics in the culture media carrying 2 marks? What is the use of HAT medium? State the components of it carrying 2 marks. Why is chromatography used in downstream processing carrying 2 marks? State the characteristics feature of type 1 and type 2 restriction endonucleases carrying 4 marks. Cite one example of each carrying 2 marks. What is meant by insertion vector and replacement vector carrying 2 marks? Distinguish between F plasmid and R plasmid carrying 2 marks. Write notes on GMO carrying 5 marks. P. Beer 322 carrying 5 marks. Liposuction carrying 5 marks. Southern blotting carrying 5 marks. What are monoclonal antibodies carrying 2 marks? State the application of monoclonal antibodies in disease diagnostic and therapeutics carrying 3 marks. What is cryopreservation carrying 2 marks? What are the principles and methods of cryopreservation carrying 3 marks? Next question. State the principle and process involved in Western blotting 2 plus 4. Distinguish between natural and synthetic media carrying 4 marks. Compare the use of phage vectors and cosmids 3 plus 3. Describe the production of recombinant human growth hormone carrying 4 marks. Describe the dye exclusion test of cell viability and MTT based cytotoxicity assay of cell survival 3 plus 3. State the principle of RAPD carrying 4 marks. State the importance of stem cell carrying 3 marks. What is homopolymer tailing carrying 2 marks? Differentiate between shuttle and expression vectors carrying 2.5 marks. Endonuclease and exonuclease 2.5 marks. Spray drying lyophilization carrying 2.5 marks. So this is all about animal cell biotechnology paper of 2022 of University of Calcutta. So students, stay tuned for next session.